Introduction to bioprocess. Bioprocess or fermentation has a long history. People started to use this technique decades ago. Back in 10,000 to 5,000 before century, people started to brew grape to be used in the beer and winemaking in the Middle East country. Year onwards, many foods were being produced using this technique such as yeast, cheese, vinegar, and even sauce. The major advance in bioprocess development occurred during World War II. They produced a large scale of penicillin. This medicine has been used to combat infection in the soldiers. Nowadays, bioprocess is widely used in all fields of study, including in the drug industry or the food industry.
In industry, everything needs to be in a huge amount. Same goes to protein production. Protein here include antibodies, enzymes, and other. So, how does company produce this thing in a bulk? They are using bioprocessing. Bioprocessing is defined as any process that uses complete living cell to obtain desired product. Bioprocessing divided into 3 stages, which is the first stage is upstream, second stage is bioprocess, and the third stage is downstream. But first, we are going to focus on stage 1, which is upstream process. Upstream process is the first step in which microbes are being cultured in bioreactors to produce a massive amount of substance. Upstream process involve first inoculum preparation, second media development, third scale-up process, and the fourth inoculation. In inoculum preparation, first we are going to have screening and selection of strain culture, second genetic modification, and third preparation of pure culture. Second process in upstream stage is media development and optimization of growth parameters. The medium used for fermentation may be classified into 3, which are defined, complex, or technical medium. Defined medium consists only of precisely chemically defined substrate. Meanwhile, complex medium is composed of substrate with undefined composition such as extract or hydrolysate from waste product, which are cheap substrate commonly used in industrial production. On another hand, technical media are used on an industrial scale and are cheaper. Next step is we are going to scale up the entire process. Scale up stage include first shake flask experiments, second laboratory stir fermenter, which is small bioreactor, and the third one is pilot scale fermenter, which is used in industrial bioreactor. The last one is inoculation. Inoculation is the transfer of seed material or inoculum into the fermenter. Once they reach the desired density, they are harvested and moved to the downstream section of the bioprocess, which is going to be explained in the next stage.
Stage 2, bioprocess. Bioprocess is a fermentation process where organisms such as microbes degrade carbon compounds with or without oxygen. The difference between bioreactor and fermenter. Bioreactors use equipment that help the maximum development of organisms, while fermenters use equipment to control surrounding conditions. Fermentation processes include batch culture fermentation, continuous culture fermentation, and fed batch culture fermentation. Batch fermentation is an enclosed system where fresh nutrients and substrates are added to a fixed volume medium and left for processing on its own. The microbes will gradually ingest the nutrients available. When the process produces the right amount of product, it is then extracted while others are thrown away. The process is done in a standard laboratory for a controlled amount, as it is not suitable for a larger spectrum. Continuous fermentation is when the medium is continuously added into the chamber. This elongates the exponential phase of the microbial growth. Continuous flow of culture equals continual removal of waste of gradual microorganism growth. It results in a maximum growth rate due to the high productivity, and there are two procedures used. The first procedure is chemostat. Chemostat is when the amount of output from the tank equals the amount of input into the tank. When the culture is added and the end product is removed constantly at the same pace, the overall volume will be similar. The second procedure is turbidostat. When the turbidity changes, it is detected by a type of sensor and the amount of input is then supervised. Thus, the output depends on the turbidity. It uses a spectrophotometer to calculate the optical density. Fed-back fermentation is where the nutrients and substrates are added constantly in the beginning until the maximum liquid capacity. It is then added at intervals, also known as controlled feeding. Then the inoculum will be added when the microbes reaches its exponential growth phase. It can be controlled with or without fed-back. Control with fed-back has a monitoring process parameters to identify concentration of substrate and product. Control without fed-back are nutrients added through controlled feeding. It needs materials like a reservoir to contain the culture and pumps for the transfer of the medium.
The important structure of a bioreactor are motor, agitator shaft, impeller, purger, cooling jacket, pH probe, oxygen probe, and also foam probe. There are 6 bioreactor types. Stirred tank bioreactor. This bioreactor type is the most widely used bioreactor. The mixing method is by mechanical agitation, and the applications include production of antibiotics. Air-lift bioreactor. The mixing method of this bioreactor type are airlift. It works by using upflowing and downflowing stream. It is suitable for fermentation of animal and plant cell cultures and also widely used for large-scale production of monoclonal antibodies. Packed bed bioreactor. A nutrient broth flows continuously over the immobilized biocatalyst. The products obtained in a packed bed bioreactor are released into the fluid and removed. These bioreactors are preferred for wastewater treatment, biological filters in small aquarium water recycling systems, and production of amino acids and enzymes. Bubble column reactor. Mixing method for these bioreactors is gas sparging, and this reactor type is the simplest design among all 6 known bioreactors and are easy to construct. They do not require agitation. Bubble column reactors are used for continuous fermentation of beer and yeast. Trickle bed reactors. In this reactor type, liquid is sprayed onto the top of the packing and trickles down through the bed in small rivulets. In the process, the gaseous pollutants in the surface of the carriers is absorbed and immediately biologically mineralized. These reactors are used for wastewater treatments. Fluidized bed reactors. In these reactors, when the packed beds are operated in upflow mode, the bed expands at high liquid flow rates due to upward motion of the particles. These reactors are used for wastewater treatments.
So, what is downstream bioprocess? The downstream part of a bioprocess refers to the part where the cell mass from the upstream are processed to meet purity and quality requirements. The volatile product can be separated by distillation of the harvested culture without pre-treatment. Distillation is done at reduced pressure at continuous yield. The steps of downstream processing are: the first step is cell removal. Separation of whole cell and other insoluble ingredient from the culture broth. Separating the biomass generally carried out by centrifugation or ultracentrifugation. If the product is biomass, then it is recovered for processing and spent medium is discarded. If the product is extracellular, the biomass will be discarded. Methods of cell removal are flotation, flocculation, filtration, and centrifugation. The second step is cell disruption. If the desired product is intracellular, the cell biomass can be disrupted so that the product should be released. The solid liquid is separated by centrifugation or filtration and cell debris is discarded. There are several biotechnological products such as vitamin and enzyme which are located within the cells. Such compounds have to be first released for their further processing and final isolation. The microorganism or other cells can be disintegrated or disrupted by physical, chemical, or enzymatic method. For physical method, there are ultrasonication, osmotic shock, heat shock, high pressure homogenization, and grinding with glass feed. For chemical method, there are alkalis, organic solvent, and detergent. And lastly, for enzymatic method, there are lysozymes and glucanase, mannase, and protease.
Then we go to the next stage in the downstream processing, which are primary isolation. What are the primary isolation in the downstream process? Primary isolation is one of the wide techniques that are available for fermentation products. It also a removal of components that properties markedly from desired product. Isolation steps are required to remove the product and reducing the volume. Adsorption, liquid extraction, ultrafiltration, precipitations are the example of the unique operations that are involved. And the next step in downstream processing are purification. Purification is the process to separate the product from impurities with similar properties. The contaminants are separated will resemble the product very closely in physical and chemical way as the significant fractions of the entire downstream processing expenditure. The example of operations are chromatography, ultrafiltration, and fractional precipitation. The last stage for the downstream bioprocess are final isolation. The final processing step which end with packaging of the product in form that is stable, easily transportable and convenient. It also removes or deactivates trace contaminants which might
comprises product safety. It usually depending on the product and its intended use. Crystallization, centrifugation or filtration, and drying are the examples of operation used in pharmaceuticals.
Next, we will learn what are industrial products involved in bioprocess.
The product includes biomass. Biomass refers to the mass of living organisms, including plants, animals, and microorganisms. For example, yeast biomass production using Saccharomyces cerevisiae. Around 0.4 million metric tons of yeast biomass, including 0.2 million metric tons packaged yeast, are produced each year worldwide.
Metabolite is produced in the industrial bioprocess through the process of fermentation by using microorganisms. There are two types of metabolites, which are primary metabolites and secondary metabolites. For example, primary metabolites such as ethanol is produced during the microbial fermentation by using E. coli. Organic acid such as citric acid is a primary metabolite which produced by microbial fermentation using Aspergillus niger. Amino acids such as L-glutamate and L-lysine are produced as the product of the industrial bioprocess. Other product is secondary metabolites. For example, antibiotics such as erythromycin. Next is therapeutic proteins. Therapeutic proteins can be used to replace a protein that is abnormal or deficient in a particular disease.
Next, enzymes. Bioprocesses, especially fermenters, are the best possible process to use or produce enzymes. Enzymes are produced from the microbes in fermenter, and the major producer of industrial enzymes is Novozymes. There are known industrial enzyme producers such as Aspergillus niger, Aspergillus oryzae, and Trichoderma oryzae. Next, food. For example, yogurt and cheese. Yogurt is produced by bacterial cultures Lactobacillus bulgaricus and Streptococcus thermophilus. And for the cheese making, lactic acid bacteria such as Lactococcus lactis, Streptococcus salivarius, and Lactobacillus helveticus were used. But it's not just it. Yeast and molds are also used in some cheese to provide the characteristic color and flavors for the cheese varieties. One of the main products from agriculture is biopesticides. There are two types of biopesticides, which are microbial pesticides and semiochemicals. Microbial pesticides are pesticides that contain living microorganisms such as bacteria, fungi, viruses, and related organisms and associated metabolites or byproducts that are used to control pests.
Semiochemicals, meanwhile, are message-bearing chemicals produced by an organism that causes a behavioral response in another organism of the same or different species. Some of the beverages are also involved in bioprocess technology. Such example, wine. During fermentation, yeasts transform sugars present in the juice into ethanol and carbon dioxide as a byproduct. The most common genera of wild yeasts found in winemaking include Candida and Zygosaccharomyces. Probiotic drink. An example of the most famous probiotic drink is Yakult. In the process of fermentation, the Lactobacillus casei shirota strain produces lactic acid from the breakdown of lactose, the predominant form of carbohydrate in milk.